Hello everyone. In previous lecture we have studied or started the evaluation of parenteral preparations. So in previous lecture we studied the two types of tests which are done for the evaluation of parenteral preparations, that are leakage test and the clarity test. So now in this lecture we will continue with the next type of test, that is pyrogen test. So all of you know that parenteral preparations, these are the sterile preparations which are free from all types of microorganisms as well as pyrogens. Okay. So it is necessary to do the pyrogen testing of each type of parenteral preparation to ensure that they are free from any type of pyrogens. So first of all we will see what are that pyrogens. So pyrogen is a Greek word which is derived from two words, that is pyro means fire and gen means it is the beginning. So pyrogens, these are fever producing, or if the pyrogens are entered in the body of human, then they produces the fever or the increases or rises the temperature of body. So these are the fever producing and these are the metabolic byproducts of microbial growth and death. Basically pyrogens, these are the endotoxins. Okay, all of you know the endotoxins. These are the metabolic byproducts which are present within the body of microorganisms, specifically gram-negative bacteria. When that particular bacteria get destroyed or after death, that endotoxins come out of the body of that bacteria. Okay. बेसिकली दोन टाईपचे टॉक्झिन्स आहेत, एक्झोटॉक्झिन्स आणि एंडोटॉक्झिन्स. एक्झोटॉक्झिन्स मीन्स व्हॉट, विच आर लिबरेटेड आउट ऑफ द बॉडी ऑफ बॅक्टेरिया. जे बॅक्टेरिया टॉक्झिन्स बॉडीच्या बाहेर लिबरेट करतात किंवा एक्सक्रीट करतात त्यांना आपण काय म्हणतो, दिज आर द एक्झोटॉक्झिन्स. आणि एंडोटॉक्झिन्स मीन्स व्हॉट, द टॉक्झिन्स विच आर प्रेझेंट विदिन द बॉडी ऑफ बॅक्टेरिया अँड आफ्टर डिस्ट्रॉईंग or when the bacteria get destroyed, that particular endotoxins come out from the body of bacteria. So pyrogens, these are the metabolic byproducts or these are the endotoxins which are lipopolysaccharide in nature, and basically they are produced from the gram-negative bacteria. So bacterial pyrogens are called endotoxins, and the gram-negative bacteria produces the more potent endotoxins than the gram-positive bacteria and the fungi. And endotoxins are heat-stable lipopolysaccharides present in the bacterial cell wall. Okay, I will just show you the structure of pyrogens. Okay, so pyrogens, these are the metabolic byproduct of bacteria, that is gram-negative bacteria. These are the endotoxins and causes rise in body temperature upon its injection. Okay. So this is the structure of pyrogen. As I said, pyrogens, these are the lipopolysaccharides. Okay, you can see here the gram-negative bacteria, and here it is a structure of cell wall of the gram-negative bacteria, which consists of three parts again, that is lipid layer known as lipid A, then there is a core polysaccharide layer, and another one is the O antigen. So it consists of three regions called lipid A, then core polysaccharide and the O specific side chain, and that particular lipid A region is responsible for rise in the body temperature and polysaccharide region increases its solubility in the water. Okay, that particular lipid A region is responsible for the rise in body temperature upon their injection. Okay, so pyrogens, these are the metabolic byproducts of, or these are the endotoxins produced by gram negative bacteria which are lipopolysaccharide in nature, and again these are the heat stable and they are generally present in the bacterial cell wall. So they are stable to at least 175 degrees Celsius. So in that case, steam sterilization is also ineffective. Okay, that particular pyrogens cannot be destroyed by steam sterilization. For steam sterilization, we are using autoclaving method, at which about 121 degrees Celsius temperature is used to kill the microorganisms. So at that temperature also these pyrogens are not destroyed. So steam sterilization is also ineffective method for the pyrogens because they are stable at least at 175 degree Celsius. Again they are water soluble and the monomer unit of lipopolysaccharide can be 10,000 Daltons, that is about 1.8 nanometer. So endotoxins can be easily passed through 0.22 micrometer filter. Again, filtration method of sterilization is also ineffective to remove the pyrogens. So mainly these pyrogens are entered via different sources like water, which is again a main source of pyrogens. Then raw materials, different types of excipients, raw materials used to prepare the parenterals, then different equipments, then process environment, then people and the protein if using gram negative bacteria. Okay, all types of vehicles like water, then different excipients, ingredients, equipments, environment, then people which are working in the parenteral area, these are the possible sources of contamination of pyrogens. Okay, then next we will see the biological property of these endotoxins, that is pyrogens. Okay, so pyrogen elevate the circulatory levels of inflammatory cytokines, which may be followed by fever, blood coagulation and the hypertension. As you know that inflammation is a defense mechanism. If any foreign particles get entered in our body, then it may causes, or it leads to the inflammation because of the secretion of different cytokines, and which may again lead to the body temperature or the fever or the blood coagulation or the rise in blood pressure, that is reducing the blood pressure, that is hypotension. If the pyrogens are entered in the body at a low dose, then they leads to the asymptomatic inflammation reaction. Okay, pyrogens जर का लो क्वांटिटीमध्ये बॉडीमध्ये एंटर झाले असतील तर ॲज सच आपल्याला कोणतेही सिम्पटम्स त्यातून दिसत नाहीत किंवा असिम्प्टमॅटिक इन्फ्लामेशन रिॲक्शन तिथे ऑकर होते. अगेन ॲट द मॉडरेट डोसेस इट मे प्रोड्युसेस फीवर and changes in the plasma composition, and at high doses they may lead to the cardiovascular dysfunction, then vasodilatation, vasoconstriction, then endothelium dysfunction and the multiple organ failure and finally death. ओके, पायरोजनची किती क्वांटिटी आपल्या बॉडीमध्ये एंटर झाली आहे, डिपेंडिंग ऑन दॅट आपल्याला डिफरंट रिस्पॉन्सेस इथे दिसतात. जर का अगदी कमी क्वांटिटीमध्ये पायरोजन एंटर झाले असतील तर कोणतेही सिम्पटम्स दिसत नाही आहेत. मॉडरेट डोसेसला फिवर किंवा चेंज इन प्लाझ्मा कंपोजिशन, अँड ॲट हायेस्ट लेवल किंवा हाय डोसेसमध्ये जर का पायरोजन्स आपल्या बॉडीमध्ये एंटर झाले तर आपल्याला डिफरंट ॲडवर्स रिॲक्शन्स इथे दिसतात. लाईक कार्डिओवॅस्क्युलर डिसफंक्शन असेल, वॅसोडायलेटेशन असेल, वॅसोकॉन्स्ट्रिक्शन असेल किंवा मल्टिपल ऑर्गन फेल्युअरसुद्धा होऊ शकतो आणि फायनली डेथसुद्धा होऊ शकते. सो टू अवॉइड दीज ॲडवर्स रिॲक्शन्स द पॅरेंटल शुड बी फ्री फ्रॉम पायरोजन्स. त्यामुळे आपले पॅरेंटल प्रिपरेशन जे काही आहेत ते पायरोजन फ्री असले पाहिजेत. ओके, सो टू चेक द पायरोजेनिसिटी, टू चेक द absence or presence of pyrogen, the pyrogen testing is done. त्यासाठी आपण काय करतो, पायरोजन टेस्टिंग केली जाते. सो बेसिकली पायरोजन दीज आर द बॅक्टेरियल एंडोटॉक्झिन्स विच आर लिपोपॉलिसॅकराइड इन नेचर and these are the metabolic byproducts of the bacteria. They are heat stable and the water soluble, and if they are entered in our body they causes rise in body temperature or they produces fever. ओके, मेन काय आहे, जर काही पायरोजन्स आपल्या बॉडीमध्ये एंटर झाले तर काय होतं आहे, बॉडी टेम्परेचर राईज होते किंवा फिवर प्रोड्यूस केला जातो आहे. सो टू अवॉइड दिस जे काही पॅरेंटल प्रिपरेशन आहेत ते शुड बी फ्री फ्रॉम पायरोजन. सो टू चेक द प्रेझेन्स ऑर अब्सेन्स ऑफ पायरोजन द पायरोजन टेस्टिंग इज डन फॉर द पॅरेंटल प्रिपरेशन. Basically these pyrogens can be eliminated from parental preparation by the different mechanisms like dry heat sterilization, okay, for glasswares, then metal equipments, powders, waxes, oil, heat stable drugs. The dry heat sterilization is done to remove the pyrogen. As we have seen the different sources of pyrogen like excipient, water, equipments, environment. Okay, these are the possible sources of contamination of pyrogen. So before producing or before processing of the parental preparation, we have to sterilize all the excipients, water, equipments, okay, to remove the pyrogens. So for glasswares, then metal equipments, powders, waxes, oil and heat stable drugs, the dry heat sterilization process is done. So in this sterilization, all these equipments are subjected to 650 degrees Celsius temperature for 1 minute or 250 degrees Celsius temperature for 30 minutes or 180 degrees Celsius temperature for 240 minutes. Again the ultrafiltration can be done. Then reverse osmosis process is also done. So reverse osmosis membrane is composed of cellulose acetate phthalate or the polyamide, or the distillation process is done, or the adsorption method is done to remove the pyrogens. Okay, so we have seen what is pyrogen, what are the different biological properties of the pyrogens and how they are eliminated by the different types of sterilization. So the pyrogen testing is done by two methods, that is by using animal in which the rabbits are used and by using the reagent like Limulus amebocide lysate. ओके, पायरोजन टेस्टिंग हे दोन मेथडनी केलं जातं. फर्स्ट आहे मेथड बाय युजिंग ॲनिमल, इन दॅट रॅबिट्स आर यूज or the pyrogen testing is done on rabbits. Another test is by using reagent. In that case, Limulus lysate reagent is used. So we will see these methods one by one. So first test is the rabbit pyrogen test. So why rabbits are used for this testing? Because rabbits are used to perform this test because their body temperature increases when pyrogen are introduced into their bodies by parenteral route. As like human rabbit also shows same responses to the pyrogen. जसं की आपण बघितलं, जर का ह्युमन बॉडीमध्ये पायरोजन इंट्रोड्यूस झाले किंवा इंजेक्ट केले गेले तर काय होतं आहे, फीवर प्रोड्यूस होतो आहे. सेम रॅबिटसुद्धा काय करतात, रॅबिटमध्ये जर का पायरोजन इंट्रोड्यूस केले गेले तर दे ऑल्सो शोज द राइज इन बॉडी टेम्परेचर और ऑल्सो दे ऑल्सो शोज द फीवर कंडिशन. सो फॉर पायरोजन टेस्टिंग for animal study, rabbits are used. So in this testing, the 3 healthy adult rabbits of either sex, each weighing not less than 1.5 kg, are selected. For this testing, initially 3 rabbits are used which has weight about 1.5 kg, not less than 1.5 kg. ओके, मिनिमम 1.5 kg वेट असणारे 3 रॅबिट्स आपल्याला इथे यूज करायचे आहेत. ऑल्सो डू नॉट यूज एनी रॅबिट हॅविंग अ टेम्परेचर हायर दॅन 39.8 डिग्री सेल्सिअस. इफ एनी रॅबिट हॅविंग इट्स बॉडी टेम्परेचर higher than 39.8 degrees Celsius, then that rabbit is not used for this testing. Again, the rabbit showing temperature variation greater than 0.2 degrees Celsius between 2 successive reading in the determination of initial temperature. रॅबिटचं इनिशियल बॉडी टेम्परेचर चेक करत असताना जर का 2 सक्सेसिव्ह रिडिंग्समधला डिफरन्स हा 0.2 डिग्री सेल्सिअसपेक्षा जास्त येत असेल तर असे रॅबिट्स आपल्याला इथे यूज करायचे नाही. अगेन, शाम टेस्ट or it is the preliminary test is performed within 7 days of the actual test. Okay, so first we will see what is that preliminary test or what is that sham test. Okay, so first one is a preliminary test. The intravenous injection of sterile pyrogen free saline solution is used. So to exclude any animal showing an unusual response to the trauma of the injection. Okay, so in this preliminary test, the pyrogen free saline solution is injected to the multiple rabbits and the responses are taken. If that rabbits are showing the temperature differences more than 0.6 degrees Celsius, then that particular rabbit is not used to done the main testing. Okay, तर ते प्रिलिमिनरी टेस्ट कशासाठी केली जाते तर रॅबिट सिलेक्ट करण्यासाठी. आपला बेसिक क्रायटेरिया काय आहे रॅबिट सिलेक्ट करण्याचा? द रॅबिट शुड वेट नॉट लेस दॅन 1.5 kg. 1.5 kgपेक्षा त्यांचं वेट कमी नसावं. अगेन 39.8 डिग्री सेल्सिअसपेक्षा टेम्परेचर त्यांचं जास्त नसावं. ओके, देन नेक्स्ट क्रायटेरिया इज व्हॉट? जर का when we inject the pyrogen free solution to the rabbit, if they are showing temperature change more than 0.6 degrees Celsius, mean they can give the false positive reading also. So for that case, to exclude any rabbit which shows the unusual responses, we are doing the preliminary test. So in this case, first the initial temperature is taken, after that the pyrogen free saline solution is injected to the rabbit, and after that again reading are checked or body temperature is checked. If the temperature difference between initial temperature and after test is greater than 0.6 degrees Celsius, then that rabbits are excluded or they are not taken for the performing main testing. So this is a preliminary test or the sham test. Okay, so animals showing temperature increase over 0.6 degrees Celsius should be removed from pyrogen testing. So after that the main testing is done on the selected 3 rabbits. Okay, so now we will see the method. In this method, first we have to prepare the solution which is to be injected to the rabbit. So dissolve the substance being examined or dilute it with a pyrogen free saline solution. Okay, the substance under testing is diluted with the pyrogen free saline solution. So that particular solution is heated or the warm the liquid being examined to approximately 38.5 degrees Celsius temperature before injection. That particular diluted solution is warm at the temperature of 38.5 degrees Celsius. The volume of injection is not less than 0.5 ml per kg and not more than 10 ml per kg of the body weight. Okay, तर आता आपल्याला इंजेक्शन ॲक्च्युली किती ml द्यायचं आहे तर नॉट लेस दॅन 0.5 ml पर के जी. त्या रॅबिटचं जे काही वेट आहे त्या वेटच्या पर के जी आपण 0.5 ml पेक्षा कमी द्यायचं नाही आहे आणि पर के जी 10 ml पेक्षा कमी द्यायचं नाही. तर या रेंजमध्ये आपल्याला तो व्हॉल्यूम सिलेक्ट करायचा आहे. अगेन विथहोल्ड द वॉटर ड्युरिंग टेस्ट. हे टेस्टिंग साधारणतः 3 तास आपल्याला 3 ते 4 तास करायचं आहे. सो ड्युरिंग दिस टेस्टिंग त्या रॅबिटने आपण वॉटर द्यायचं नाही आहे किंवा वॉटर आपल्याला स्टॉप करायचं आहे, विथहोल्ड करायचं आहे. सो क्लिनिकल थर्मामीटर इज इन्सर्टेड इन टू द रेक्टम ऑफ रॅबिट टू रेकॉर्ड द इनिशियल बॉडी टेम्परेचर. ओके, फर्स्ट आपण काय केलं, टेस्ट सोल्युशन तयार करून घेतलं. ते कसं केलं, ज्या कोणत्या पॅरेंटल प्रिपरेशनचं किंवा सबस्टन्सचं आपल्याला पायरोजन टेस्टिंग करायचं आहे तो आपण पायरोजन फ्री सलाईन सोल्युशनसोबत डायल्यूट करायचं आहे. ओके, डायल्यूट मीन्स डायल्यूट मीन्स व्हॉट, इट्स अ 5% सोल्युशन. सो ओके, सो हे तयार झाल्यानंतर आपल्याला काय करायचं आहे, ते सोल्युशन 38.5 डिग्री सेल्सिअसपर्यंत वॉर्म करायचं आहे आणि ते वॉर्म सोल्युशन आपल्याला रॅबिटना इंजेक्ट करायचं आहे. तर आता हा व्हॉल्यूम किती इंजेक्ट करणार आपण, नॉट लेस दॅन 0.5 ml अँड नॉट मोर दॅन 10 ml पर के जी ऑफ दॅट रॅबिट. ओके, ऑल्सो withhold the water intake of that rabbit. आफ्टर दॅट ॲक्च्युअल टेस्ट करत असताना आपल्याला काय करायचं आहे, बिफोर इंजेक्शन वी हॅव टू टेक द इनिशियल बॉडी टेम्परेचर ऑफ दॅट रॅबिट. पर्टिक्युलर इंजेक्शन देण्याच्या आधी आपल्याला काय करायचं आहे, तर रॅबिटचं इनिशियल बॉडी टेम्परेचर किती आहे ते चेक करायचं आहे. सो फॉर दॅट द रेक्टल थर्मामीटर्स आर यूज. Okay, that particular thermometer is inserted into the rectum and rectal temperature is taken. So for that 2 normal readings are taken at the interval of half an hour and its mean is calculated as an initial temperature. Okay, काय करायचं आहे आपल्याला, रेक्टल थर्मामीटर यूज करून बॉडी टेम्परेचर चेक करायचं आहे. असे आपण 2 रिडिंग्स घेतो आहे अर्धा तासाच्या इंटरवलने आणि त्या 2 रिडिंगचं आपण मीन काढायचं आहे आणि ते आपलं काय झालं, इनिशियल बॉडी टेम्परेचर ऑफ दॅट रॅबिट. ओके, आफ्टर टेकिंग द इनिशियल टेम्परेचर पर्टिक्युलर टेस्ट सोल्युशन इज इंजेक्टेड टू द रॅबिट. ओके, द सोल्युशन अंडर टेस्टिंग इज इंजेक्टेड थ्रू अन ईयर वेन. Again, record the temperature of each rabbit in an interval of 30 minutes for 3 hours. Okay, टेस्ट सोल्युशन रॅबिट्सना इंजेक्ट करायचं आहे थ्रू द इयर व्हेन. अगेन द टेम्परेचर readings are or the temperatures are recorded at the interval of 30 minutes for 3 hours. म्हणजे आपल्याला 3 तासामध्ये 6 रिडिंग घ्यायचे आहेत अर्धा तासाच्या इंटरवलने. Again, the difference between initial temperature and the maximum temperature is recorded and taken as the response. ओके, जे आपण इंजेक्शन दिल्यानंतर 6 रिडिंग्स घेतोय त्या 6 रिडिंगमधलं मॅक्झिमम टेम्परेचर किती आहे ते आपल्याला कन्सिडर करायचं आहे आणि त्यातून आपल्याला इनिशियल टेम्परेचर जे काही होतं इंजेक्शन द्यायच्या आधी ते मायनस करायचं आहे and that particular temperature difference is recorded as a response. Okay, response of that particular rabbit. So next one is the interpretation of result. How the results are interpreted. For 3 rabbits, its individual temperature rise should not be more than 0.6 degree Celsius. प्रत्येक रॅबिटचा टेम्परेचर डिफरन्स जो काही आपण घेतला आहे तो 0.6 डिग्रीपेक्षा जास्त नसावा किंवा इन ग्रुप द टेम्परेचर डिफरन्स should not be more than 1.4 degree Celsius, then the test passes. ओके, 3 रॅबिट्सचा प्रत्येकाचा बॉडी टेम्परेचर जो काही टेम्परेचर डिफरन्स आपण कॅल्क्युलेट केला आहे तो 0.6पेक्षा जास्त नसावा किंवा ग्रुपमध्ये तिघांचाही 1.4पेक्षा जास्त टेम्परेचर डिफरन्स नसावा तर आपली टेस्ट ही पास होऊ शकते. इफ अबाव टेस्ट नॉट पासेस. ओके, जर का फर्स्ट 3 रॅबिटवरची टेस्ट पास नाही झाली तर अगेन दॅट पर्टिक्युलर टेस्ट इज डन ऑन मोर 5 रॅबिट्स. आणखीन 5 रॅबिट्सवरती आपल्याला ती टेस्ट करायचं आहे आणि टोटली 8 रॅबिटचे रिडिंग आपल्याला इंटरप्रेट करायचं. अगेन सेम, द इंडिव्हिज्युअल टेम्परेचर शुड नॉट बी मोर दॅन 0.6 degree Celsius and in group the temperature difference should not get more than 3.7 degree Celsius, then the test passes. If above test not passes, perform the test again. 8 रॅबिटवरची टेस्टसुद्धा फेल झाली तर पुन्हा टेस्ट आपल्याला करायची आहे. अँड इफ अबाव द टेस्ट नॉट पासेस द सॅम्पल इज सेड टू बी पायरोजेनिक और गो थ्रू द सोर्सेस ऑफ कंटॅमिनेशन ऑफ पायरोजन. अगेन रिटेस्टिंगसुद्धा फेल झालं तर पुन्हा आपण काय म्हणू शकतो की पर्टिक्युलर जे काही टेस्ट सोल्युशन आहे इट इज अ नॉट स्टराईल और इट इज अ पायरोजेनिक. सो यू हॅव टू गो फॉर or you have to find the sources of contamination of pyrogen. Okay, so this is the animal test which is done on the rabbits. The next test is the reagent test or the bacterial endotoxin test, or we can say it as a LAL test, that is Lumulus amoebocyte lysate test. Okay, so in this test, to detect or quantify the endotoxins of gram negative bacteria origin. Okay, so this test is done to quantify the endotoxins of gram negative bacteria. So in this case, the reagent like amoebocyte lysate enzyme, which is extracted from horseshoe crab, that is Limulus polyphemus or Tachyphyllus tridentatus species. Okay, so second test is a reagent test, or we can say it as a LAL test, that is Limulus amoebocyte lysate. So this amoebocyte lysate is a type of enzyme which is extracted from the horseshoe crab. So horseshoe crab is a species of marine or brackish water arthropod. Horseshoe crab, these are the type of arthropods which are found in the marine water or the brackish water. Okay, horseshoe crab हे काय आहेत? These are the type of arthropods or these are the invertebrate found in the marine water or the brackish water, and that particular amebocyte lysate enzyme is extracted from the blood cells of the horseshoe crab. ओके, त्यांच्या ब्लड सेल्समधून अमिबोसाइड लायसेट हे एन्झाईम एक्स्ट्रॅक्ट करायचं आहे, अँड दॅट पर्टिक्युलर एन्झाईम इज यूज ॲज अ reagent for doing the LAL test. So it has a simple mechanism. The test is based on the gelling property of the enzyme extracted from the horseshoe crab of Limulus polyphemus, and that enzyme, when come in contact with the bacterial endotoxin, the gelling occur, and the degree of gelling related to the amount of endotoxin present. Okay, so ameboside लायसेट हे एन्झाईम आहे की जे हॉर्सशू क्रॅबच्या ब्लड सेल्समधून एक्स्ट्रॅक्ट केलं जातं. वेन दॅट पर्टिक्युलर एन्झाईम कम इन कॉन्टॅक्ट विथ द बॅक्टेरियल एंडोटॉक्झिन दॅट इज पायरोजन, द जेलिंग ऑकर. ओके, जेव्हा हे एन्झाईम बॅक्टेरियल एंडोटॉक्झिन किंवा पायरोजनच्या कॉन्टॅक्ट्समध्ये येतात तेव्हा त्यातून काय होतं, जेल फॉर्म होतं आहे, अँड दॅट डिग्री ऑफ जेलिंग इज related to the amount of endotoxin present in the particular solution. So how this test is performed? So to avoid the endotoxin contamination, the interfering factor should not be present. Again, the equipment should be depyrogenated. The sensitivity of the lysate should be known. The equipments or the apparatus which are used to do this testing, they should be pyrogen free. Then how this actual testing is done? The equal volume of LAL reagent, that is Limulus ameboside lysate reagent, and the test solution, usually 0.1 ml of each, are mixed in a depyrogenated test tube. Okay, so we have to mix LAL reagent 0.1 ml with the 0.1 ml test solution in a depyrogenated test tube. After that, incubation is done at 37 degrees Celsius for 1 hour. That particular test tubes are incubated at 37 degrees Celsius for 1 hour. Again, remove the test tube and invert at a 180 degree and observe the result. Okay, so I will just show you one diagram from which you may get a clear idea of this testing. Okay, so in first test tube, the 0.1 ml LAL reagent is mixed with the 0.1 ml test solution. After incubation of 1 hour, the test tube is slowly inverted at 180 degree. आपल्याला 180 डिग्री अँगलला ती टेस्ट ट्यूब इन्व्हर्ट करायची आहे स्लोली. इफ द सोल्युशन इन द टेस्ट ट्यूब फ्लोज, then the test passes, that is gelling is not occur. And why that gelling is not occur? Because pyrogens are absent. But if the solution in test tube does not flows when we invert the test tube at 180 degree, that means what? The gelling has occurred. And why that gelling is occurred? Because the pyrogens are present in that particular solution. And if we found the gelling in the test tube, that means our test solution fails the pyrogen test. Okay, so this is the result of this LAL test. Again, this LAL test has some advantages over the animal test, like it is a fast test, around 60 minutes or 180 minutes are required. Around 60 minutes are required for the lal test and 180 minutes are required for the rabbit test. So it is the fast test. Again, it shows the greater sensitivity. Again, there is a less variability. Not much skilled personnel are required. Again, much less false positives. There are less chances of the errors from human or the animals. Again, it is a much less expensive. It requires a low cost. Again, alternative to animal model, animals are not required, and it is a accurate testing. Okay, there are less chances of inaccuracy, less time consuming, and it gives a more accurate results. So these are the two testings which are done to check the pyrogenicity. Our pyrogen testing is done by these two testing. Again, these two tests are very important as per the exam point of view. Either one test is asked in each year question papers. ओके, या दोघांमधली एक तरी टेस्ट तुम्हाला कंपल्सरी विचारलीच जाते. ओके, सो जस्ट आपण एकदा रिवाइज करूया. Pyrogen testing is done by two types of test. First one is the animal test in which rabbits are used. Second one is the reagent test. So for rabbit test, 3 rabbits are used which weighs not less than 1.5 kg and they has a body temperature below 39.8 degree Celsius. Okay. Again, the substance under testing is diluted with the pyrogen free saline solution and that particular diluted solution is warm at 38.5 degrees Celsius. Again, how much volume is injected? Not less than 0.5 ml per kg and not more than 10 ml per kg of the rabbit. Okay, so before testing, the initial temperatures are recorded for each rabbit, and how that initial temperature are recorded? The clinical thermometer is inserted into rectum and the two readings at the interval of 30 minutes are taken and the mean of that reading is taken and it is taken as the initial temperature. After that, the solution under testing is injected through the ear vein. After injecting the solution, the 6 readings are taken at the interval of half an hour within 3 hours. Okay, and how the results are interpreted? Out of the 6 readings, the highest recorded temperature is taken and the initial temperature is compared with that particular test temperature and the difference should not be more than 0.6 degrees Celsius for each rabbit, and in group the temperature difference should not exceed 1.4 degree Celsius. If the test fails, again extra 5 rabbits are taken and testing is done on the 8 rabbits. Again, for that 8 rabbits, individual temperature of the rabbit should not be more than 0.6 degrees Celsius and in group the temperature should not be more than 3.7 degrees Celsius. Again, if the retesting fails, then one more retesting is done, and if that second retesting is failed, then we can say that our particular parenteral preparation is contaminated with the pyrogen. So we have to check the source of contamination of that particular pyrogen. Again, in reagent testing, the Lumulus amoebocyte lysate enzyme, which is extracted from blood cells of horseshoe crab, is taken as a reagent. Okay, लाल टेस्टिंग मध्ये आपण रिएजंट कोणता यूज करतोय, दॅट इज Lumulus amoebocyte lysate enzyme. तर आता हे एन्झाईम कुठून काढतो आपण? फ्रॉम द ब्लड सेल्स ऑफ हॉर्सशू क्रॅब. हॉर्सशू क्रॅब इज अ टाइप ऑफ आर्थ्रोपोड फाउंड इन द मराइन वॉटर and from its blood cells, that particular blood cell move like amoeba with a pseudopodium, so they are known as amoebocyte. And that particular from that particular blood cells, that amoebocyte lysate enzyme is extracted. And what is the principle of LAL test? When that particular amoebocyte lysate enzyme come in contact with the endotoxins of the pyrogen, it shows the gelling. Okay, so these gelling properties, so this gelling shows the presence of pyrogen. So आता प्रोसिजर कशी केली जाते याची? 0.1 ml amoebocyte lysate enzyme is mixed with the 0.1 ml test solution. That particular test tube is incubated at 37 degrees Celsius for 1 hour. After 1 hour, that particular test tube is inverted at 180 degree of angle. Okay, if the solution flows from the test tube, means what? Gelling has not occurred, means pyrogens are absent in the solution. But if the solution does not flow from the test tube, means what? The gelling has occurred. Gelling, why the gelling occur? Because the pyrogens are present in the test solution. Okay, this is a simple procedure and principle of the LAL test. Again, these two both the tests are very important as per the exam point of view. Okay, principal procedures are also easy to understand. So for any query, please ask to me, and thank you for your attention. Okay, thank you.